Hello, and welcome to Bioinformatics. My name is Jeff Damath. I am going to be the instructor for the course. I'm an associate professor and associate chair in biology, and I've been teaching this course for about 14 years. This is one of my favorite courses to teach, partly because it's a field that moves really rapidly, and so across those 14 years, there's almost nothing, really, there is nothing that has stayed the same since the first time I taught this course. Used to the coding language has changed. Most of the topics we've changed, the approaches, just really fun for me to teach, but also fun because it's a very practical course. You guys are going to pick up a lot of skills that are super high demand in terms of lots of wide selection of the workforce. And so that part is exciting as you sort of build these computational skills that let you approach biology questions from a computational perspective. Obviously, we're going to get into that a lot more as we go through the semester. The purpose of this video is really to introduce the course. Like you, we're going to go over the syllabus, and I'm going to go over the Canvas shell and sort of orient you to how the course is laid out in Canvas. So with that said, let's shift over and I'll share my screen. If you're watching this video, you have already made it to the home page for the course, and this video right in the middle of the page is the video that you're watching right now. So if this is your first time here, the first thing that you'll want to do, I've got a little list here, is to look over the syllabus. So if you click on this link, takes you to the syllabus page, and you can download the syllabus. Also gives you a little bit of a course summary. I'll come back to these assignments, but as the assignments get published, they'll pop up here. Probably most of you are somewhat familiar with Canvas at this point, so not too much new there. When you download the syllabus, you'll get to a document like this. This is the Word version. I think the download is a PDF. It's all the same content. So this is me. We've got two graduate teaching assistants that I'll introduce a little more when we get back over to the Canvas shell. There's a page that has a little more info on all of us, but it's got our office locations. ERB is the Engineering Research Building. We're on the north side of campus. If you're not familiar where our offices are or where the classroom for the in-person sections, you can click on the campus map there. The best way is to get in touch with me, not by phone. This phone number will take you to the main office. I don't really answer my office phone. The best ways to get in touch with me are email or Microsoft Teams. You can call me on Teams, that's fine. Typically, I'll respond to DMs or emails pretty quickly, but certainly by the end of the next business day. If you don't hear back from me, I promise I'm not intentionally ignoring anyone. But sometimes it gets lost on the inbox page or the little red dot goes away and before I get back to it. So feel free. If I don't get back within a day or so, just send me a reminder. And then the office hours are also listed there, and those are also, you'll see those in Canvas. Course information, there are 3 sections. The undergraduate and one of the graduate sections meet on campus. The in-person meetings are Tuesday, Thursday from 2 to 3:20 in COBA 339. That's the business building. And then the online section is asynchronous. I'll get into the format of the course a lot more in a minute. But starting September 3rd, so that's the beginning of the 3rd week of class, if you're in the asynchronous online section, you are welcome to come attend the in-person section, obviously with the caveat that there needs to be space available. But typically that won't be a problem. The room that we're in this year, 339, should be plenty big for the number I anticipate to be there at any given class time. Okay, time zone, obviously that's for people who are in the asynchronous That's important. The course content, like I alluded to a minute ago, this is a very practical, sort of hands-on, skills-based class. This was designed originally and still carries a lot of that history uh, to be a course for biology students who don't have any programming background. So for those of you who are data science or maybe computer science taking this course, especially at the beginning of the course when we're doing a lot of Unix, Linux, learning Python, if you've had any kind of programming background in those using those languages, it's probably going to seem very simple. Then when we get to the second half of the class and we talk more about applications and we lean more into the biology and applying what we've learned in the first half, that's where it typically tends to flip in terms of who's having uh, more of the challenge, let's say. Um, but just recognize that this was designed to build coding skills in the first half of the class. So if you already have those coding skills, uh, the homework won't be as much of a challenge, probably. Hopefully, it'll still be fun and a little bit of a challenge, as they may be new types of algorithms and things for you to work on. But certainly a programming background, I assume you don't have any, but if you do have some, obviously that's going to give you a little bit of a leg up at early in the semester, at least. Prerequisites, introductory biology and genetics. Those are pretty loose. I think you can, if it's been a while since you had genetics or intro bio, I think you'll still be okay. Most of the biology you need, I introduce along the way. And certainly give you links and outside reading that to get you caught up if there are concepts that you are not familiar with. The learning objectives, learning outcomes for the course, there are 3 of them. You'll see down below. All of the things that we do in the course are meant to be lined up with these 3 objectives. Want you to be able to solve computational problems using Linux and Python. If you're already asking why Python, you might be a little bit you're one of those who might have some background in maybe R or something like that. I address that when we get into the Python section. Why Python, not R, for example. Second learning outcome, you'll be able to deconstruct challenges in molecular genetics. So you've got a biology question, and then we'll sort of walk through how do you turn that into, can you turn that into a computationally tractable question, and then implement some kind of algorithm to try to answer that question. And then the third one is applications, or we will learn to analyze and interpret genome and transcriptome, that is gene expression data, using both local, meaning your own machine, and remote computing resources. We'll log in and use some of the high-performance computing resources that are available through the federal government. There's no textbook for the course. Everything you need will be made available through Canvas, or I will share directories in some cases on the supercomputers that have the data. Major assignments. This is a heavy homework based, right? That's how you build skills. I will give you problems, and then you'll have time to solve those. 30% of the class, which is 1 quiz and 23 Rosalind problems. I'll talk about what Rosalind is. It's an online homework platform. There are 25 of those at 5 points apiece. I think I got the numbers wrong there. Obviously, that's 23. That should be 23. Fix that. So 24 total coding homework problems. And then there's a midterm exam that will be at the end of that coding section of the class. That is 10% of the grade. There are right now 4 computational genomics assignments. If this depends on how the pace of the course is going, whether I drop one of those or add one of those, and then the points will be adjusted accordingly so that that 120 points is split across those 3 to 5 assignments. And then finally, there will be a final project that has an analysis part, a written part, and a presentation part, and that is worth 30% of the grade. Lots more details to come on all of that stuff as we work through the semester. Expectations for out-of-class study. You should anticipate, if the standard is 3 hours outside of class for every hour in class, that's the sort of standard metric that UTA puts in. I'm saying you should expect to do at least that for coding, especially if you have never coded before. Expect this to be challenging and take time. There's no shortcut to learning a new language. Yeah, just be ready to spend time. I'm not going to sugarcoat it. Technology requirements. If you have a relatively recent laptop, you should be fine. If you're a Mac user, the terminal application on a Mac functions as a Linux terminal. You need to be in Linux. That is the world of scientific computing. There's no way around it. You have to learn it if you're going to use outside resources, the kind of resources you need in order to analyze real large genomic and transcriptomic data sets. They all are on Linux machines. So you need to have a Linux terminal. If you're on a Mac, that's easy. If you're on Windows, it gets easier and easier every year. I'm pretty sure you can go to the Microsoft App Store now, at least on my Microsoft Surface. It was very simple. Just go to the Microsoft App Store, download Ubuntu, and it installs, activates Windows Subsystem for Linux, and very easy. If that fails, so you've got a little bit older computer or your version of Windows 10 isn't up to date, there are some links here that might help walk you through that. Windows, at some point during Windows 10, Windows started shipping with something called Windows Subsystem for Linux, and you have to get that activated in order for Ubuntu to run. So there are some links here that might help you walk through that if you run into problems with that Microsoft Store download and install. These are just some links. The library does have laptops that you can check out. I think it's best if you can have your own, especially if you're learning this to do your own research or hope to do research in the future. You're going to set up a lot of software and environments that you're going to be coding in, and it's just nice to have those up and running. And there are some things that settings that you can personalize that may make life easier. And if you're having to check out or switch laptops all the time, there's a little bit more of an energy of activation if you have to do that. If you want to record classroom or online lectures, I say that's fine here. Like, this is UTA language. There shouldn't be any need for you to do that, though. Everything that you need to have for the course is already recorded and distributed through Canvas. Grading information, pretty standard. No curve. It's just the total number of points you get here. So here it's broken down by each one of the assignments. For example, Rosalind homework 1 through 9. Each one of those problems is worth 5 points. It's just that I have a single due date, so that's why they're lumped. And SLO, that's student learning outcomes. So how are the different assignments lined up with the outcomes there? Just gives you a little bit more finely grained breakdown of how the points will work out over the semester. And then standard grade calculation. I've never had to curve grades before. In this course, if you do the homework and keep up with the class, so you're turning things in on time, you will do well. That's the bottom line. If you get, for example, on all of these homeworks, you get to try them over and over until you get them right. And so as long as you start trying early enough so that you can get them right by the due date, you should get all the points. Makeup exams and late work. The homework assignments will stay open after the due date. You just get a 50% reduction in the point value. For the midterm exam and the final project, I will not accept late assignments. Okay. There's 5 points extra credit when we get to the HPC, that's high performance computing login module. That's really just to motivate you to get your credentials set up, and so that everybody can log in when we're ready to actually start running applications on the remote computers. I'll do my best to get Canvas updated whenever we've got assignments coming in. If you ever have grade grievances, those need to go through the biology department first. So some links here. Everything beyond this, university policies and all that, this is mostly boilerplate from the UTA syllabus. You'll probably get most of this in all your courses. The exception being attendance. So attending class sessions is often a good predictor of student success. It's not so much in bioinformatics. One of the things that we learned during COVID, or I learned during COVID, is that doing these video lectures is really a benefit to student learning for learning to code. So the reason I've gone to a mostly, well, really entirely in terms of the content of me giving you information is in pre-recorded lectures, because you can do them at your own pace in your own environment. And so, for example, we're going through a tutorial and whatever the exercise is doesn't work for you. Rather than getting lost in an in-person lecture as I move on to try to balance keeping the people who it worked for up to speed with helping the people who it didn't work for, you can just pause the video, right? So this online method of giving you content is really the best way to learn to code, I'm convinced. And so all that is to say, no matter what section you're in, whether you're in person or in the asynchronous online, attendance is optional in terms of in-person attendance. Everything you need for class will be provided through Canvas. And then here again, saying the beginning of the third week of class, if you're in the online section, you can come, and then the caveat being there is a finite number of seats in the room. But because it's given to you online and you can work at your own pace, if you come to class, what that means is you're going to be watching a video in class, working through the problems as you watch the video. The benefit to being in class and doing that is that myself and the TAs will be there to help you when it doesn't work. So we can help you figure out why didn't it work. An even better way to do class is to try to work through the videos before that class, then come to class with the problems that you know you're already going to have. And it's a more efficient way for us to help more people that way. And it helps you make progress faster. Okay. because, like I said, this is a skills-based course and people are coming into it with wide variation in both their biology and their computational background, the pace in the different parts of the course is highly variable among students. That's fine. It happens every semester. That's why we're here to help. That in-class Tuesday, Thursday from 2 to 3:20, it's not going to be me getting up in front of class and doing a new lecture. The lectures are recorded and available for you already. Okay, so just trying to make that expectation clear for what's going to happen during the class periods. They work just like office hours. They're there for me and the TAs to help troubleshoot any of the challenges that students are having. Okay. The generative AI use, you should use it. So this is boilerplate. UTA now has to have a policy, or instructors should have a policy for using AI in their courses. My policy is that I have unrestricted use of generative AI. So you can use ChatGPT to help you solve the homework. That's fine. This is the way the world works now. If you don't learn how to use it, you're going to fall behind in terms of the workforce and academia and everything. That said, so this is what that policy means. But note the bit in red here. Unrestricted use of generative AI does not exempt you from general prohibition against plagiarism. So the plagiarism checks still get run, and if your code or your homework gets flagged for plagiarism, that's still a problem, right? You can't just copy paste somebody else's work, even if it's a computer generated somebody, okay? So just be aware. I encourage you to learn to use ChatGPT or Claude or Gemini and any of the uh, generative AI resources to help you be more productive. But that doesn't excuse you from this prohibition against plagiarism. And then after that, the rest of the syllabus are resources. There's nothing unique to my course here. If you have any questions about that, feel free to shoot me a message, and I'll be happy to get back to you as soon as I can. The last page of the syllabus gives you a very course schedule, where just broken down by week. You'll see when we switch over to Canvas, I've got this day by day, but it gives you an idea of what we'll be covering when, and then where the assignments fall out there. Okay, so let's shift back over to Canvas now. And I'm going to go, so this is the syllabus page. I'll go back to the home page. And the number 2 on first time is module 0, the getting started module. So this takes us over to modules, and you can see the first module is laid out a little bit differently than all the other ones. So once you get to content modules, they're pretty much all laid out the same way. You have an intro page and then really the main content. But in this getting started module, it has a lot of the content that we just went over in the syllabus. So I'll go relatively quickly, but I did say I would introduce us a little bit more when we got to this point. So this is me. If you want to know the kind of research my lab does, a good way to do that would be to go over to my Google Scholar profile. That'll show you. You can sort that by the things that are the most cited from my work, or you can sort it by year and see what we've been doing recently. The most bioinformatics relevant things that we've been doing recently have been single cell transcriptomics, which we'll talk about later in the semester toward the end. Office hours, my email, the UTA faculty profile are there. and then again, my preferred methods of communication. Adam and Zoe are PhD candidates in biology. Their offices Adam is right below me in ERB, and Zoe is across the hall. We've kind of tried to spread out our office hours across the week, so hopefully uh, that gives you plenty of opportunities. And again, right, the in-class time is also maybe the best time because you can get all three of us potentially. Um, those office hours, the class time on Tuesday, Thursday from 2 to 3:20 really function like office hours. We are there to help troubleshoot as you try to go through the different exercises. Okay. So the next page in the Getting Started module, a lot of this is the same. So as in the syllabus, just copy paste it. So I won't go through that again. So it's got the attendance and expectations there. Just highlighting here the keep up, and it's worth bolding and underlining by click over to the course schedule. So especially if you're in an asynchronous class, sometimes you may put things off, and I want to encourage you up front not to do that. So this is what I meant by a more detailed schedule. So this gives you day by day, in terms of the Tuesday, Thursday class, what content you should be getting through. So when I say keep up with the content, keep up with the assignments, this is what I mean. For example, by September 5th, you should have watched the Beginning Bioinformatics Part 4 video. And you'll see when I'll show Module 1, this content corresponds with videos. So that is the name of the video with the link in Module 1 that you should have gotten through by, in this example, September 5th. And then you'll see out to the far right, if there's an assignment due that day, it's due by midnight on that day, right? So on 3rd of September, there will be a quiz that covers the Unix section of the course. And then there's another relatively large gap before you have another due date for homework. So Rosalind problems 1 through 9, but you can see in the column of goals where you should be trying to get to. So for example, on 12th of September, you should have watched that video and you should get all the way through Rosalind problem 4. So the fact that 1 through 9 are not due until the 24th of September. If you wait until September 23rd to try to do Rosalind problem 1 through 9, especially if you don't have a coding background and you're not good in Python, you won't finish. That I can almost guarantee it. So keep up with the videos and try to keep up with the goals. If you're not able to keep up with the goals, that's when you should be coming to class, the in-person Tuesday, Thursday, or coming to office hours, or both, okay? Just so that we can help you continue to make progress and keep you up to speed. Go back. So that was expectations of you, right? Keep up. And that's really for your own benefit, because this will overwhelm if you get far behind, because those are time consuming, especially as you get into the later assignments, the later homework problems in Rosalind. Okay, and then expectations that you can have of me, basically that I'll communicate in a timely way and get you your grades in a timely way. And I'll do my best to live up to all those expectations. Technology requirements, already talked a little bit about getting Linux up and ready. This top part is standard for any UTA course, I believe. The Linux, Anaconda, and Rosalind, you don't need right away, but these are here with links, and they get reintroduced when you get to that section. So the videos will have a little bit more walkthrough in terms of doing that. But the earlier, if you want to get a head start on getting things installed on your machine, there are links here for you to do that. I keep mentioning Rosalind. So this is an awesome online homework platform. It generates a data set in real time. You run your code and then upload the answer, and then it tells you whether you got it right or not. That's it in a nutshell. To get into Rosalind so that you can see the problem set, the homework problems for this course, there's two steps. First, you need to register for Rosalind. So I've given you a link to register there. Sometimes UTA's email server does not like the validation emails from Rosalind, so they get filtered and they'll either go to your spam or they won't come through at all. So it's fine if you want to use a non-UTA email to register on Rosalind. It's not being automatically linked to Canvas anyway. So it doesn't matter as long as you get registered. And then the second step is to enroll in the course. So once you have a login for Rosalind, you should follow this link, and this will take you to the appropriate section. I've got it split. Even though you have the same problems, this is just a bookkeeping efficiency for me in terms of keeping online versus in-person section people separate. So yours Canvas shell will take you to the correct enrollment link. And then once you're enrolled, you should see something like this, right? It'll say 2024 with your appropriate section, either 1 or 2, and then all the Rosalind problems with their due dates, okay? Once we get to that section, we don't get to that section until we start coding in Python, which is really the second right. First, we start with Unix, and then we'll move to Python. So you won't really need to start working in Rosalind until Python. Just a word, big picture. Once you're done with the course, I am giving you only a small sliver of the problems that are in Rosalind to do. If you're already somebody who knows how to code, I encourage you to explore all the stuff that Rosalind has. It has some very sophisticated computational challenges in there. And if you can work through more of Rosalind, all the better. It's not credit for the course, it's just skill building. But these are the kinds of skills that get you jobs and help you do research if you don't sort of specifically want to become a bioinformatics person as a career. The rest of these are university boilerplate, so nothing unique here for our course, just more information and resources that UTA provides. Again, more resources that UTA has available for you. These are more general, not specific to bioinformatics, but good to know. The academic integrity. This is another standard one, but has that bit about the generative AI use, just for those who end up skipping over the syllabus at some point. It's also here in Canvas, just so you know, you're welcome to use generative AI. Just recognize that plagiarism is not okay. And then last thing, there is a discussion board here. Historically, this is not used very often, but if you have a question for the class, for example, or something you think would benefit lots of people, you're welcome to post it here, and including questions that I will answer as I see them. I think that's it for the Getting Started module. So if you just keep clicking next, it takes you automatically into module 1. all the modules are laid out pretty much the same. So let me switch over just to show you overview again. So we just went through all the items in getting started. And then it took us over to module 1 at a glance, which has an introductory video that gives you an overview for the whole module. Going back here, gives you the objectives, and it has a to-do list and what is going to be due by the end of that module. Then if you click next, this is the video and media page. So this is really in each one of the modules where most of the content, the sort of meat of that module, and it's got all the videos that you'll need to watch. If you remember back to the course schedule, I had those names for the content, right? Those match up with these videos. So that's what you should be watching. In this module, all of those have documents that go with them. So you'll want to download the document, watch the video, which is going through the document, and sort of, at least in this module, it starts out very sort of holding your hand, walking you step by step. I'm typing it on the screen, you're typing it on your computer, and then making sure things run. Okay. And then in module 1, there's actually a quiz, and you'll want to wait and take that after you've completed all of the content. So once you get to that point in the schedule, right, and so that quiz is not due till September 3rd, in the videos, in the content, it will talk about this. So that's how all the modules are essentially laid out. Right now, only module 1 and 2 are published. That takes us all the way to mid-October, though. Let me go back to the home page where we can see the schedule. Right, the first 2 modules are learning to code, and then the second module is really algorithms. But in module 2, you're learning more Python, more coding as you learn algorithms. Those are by far the 2 longest modules. After that, we're essentially 1 week per module as we go through different bioinformatics applications and address real biological questions with real biological data as we learn to do sequence alignment and search through large databases, how we identify mutations in a genome, how we quantify gene expression. Final projects, I'll say a lot more about that later in the semester. Right now, we have some interesting and potentially exciting pathways projects that I have in mind. Every semester, we tend to do different, I give you choices of a different suite of real data to do your final project on, because I want this to be real research, right? This is, at the end of the semester, you're equipped to do some real research and potentially generate some real publication at the end of the day. We've got some new genomes and lots of new genomes and some new transcriptomes as well. And so right now it's just deciding how do we divide that up. So lots more detail to come on final projects. Yeah, I think that should give you enough of an overview, give you an idea of what's to come. If you have any questions, feel free to reach out and I'll be happy to answer those. For those that are in the in-person section, I look forward to meeting you soon. For those in the online section, hopefully you'll, even if you don't need to come to class, hopefully you will take the time, come say hi. Look forward to meeting you all, and hope you have a great semester.